Welcome to the Deep Dive. Today, we're tackling a really critical challenge in agriculture, the fungal diseases that attack maize crops.
Yes, a huge topic.
It's a major issue globally, and especially in sub-Saharan Africa, where maize is just so vital for food security. The latest FAST data really underscores that.
Absolutely. It's a cornerstone crop.
So our goal today, our mission, if you like, is to really break down these diseases. How do we spot them? What are the current ways we're managing them? We'll be digging into scientific research, things like aflatoxin, maize's own defenses, and also practical advice from agricultural guides on, say, stalk rots and leaf blights.
Sounds good. It's true. Maize faces constant threats, and fungi are definitely high on that list. The sources we've got give a pretty comprehensive picture, I think.
Yeah, from the toxins they produce right down to the plant's genetics.
Exactly how it fights back.
Okay, let's dive in. Let's start with something that's not just about yield loss, but has serious human health implications. Aflatoxin contamination.
Right, a really important one.
A CGSpace report gives us some useful insights here, focusing on Burkina Faso, Mali, and Niger. And it's not just maize, is it?
No, that's a key point. The report mentions groundnut and sorghum can also get contaminated.
So focusing on Mali for a second, there's data in Figure 2 showing aflatoxin levels in samples of groundnut, GN maize, MZ, and sorghum SG across different regions like Kays, Koulikoro, Ségou, Sikasso.
And what you see there is quite a bit of variation, isn't there?
Yeah, it really jumps out how much the levels seem to differ region to region, even for the same crop.
And Table 3 helps us compare those crops directly within regions. It shows the average concentrations.
Ah, okay.
So if you look at Sikasso, for example, the average in maize is statistically similar to sorghum, but it's different from groundnut. Those little letters next to the numbers, they tell you which averages aren't significantly different in that study.
Right, so the risk isn't uniform. Different crops, different areas, different levels.
Precisely. And it's worth noting the ND not detected. Sometimes, thankfully, it's just not there.
Good point. And we should probably mention the funding for this work came from several groups. USDA AFAS, the Norwegian Embassy in Mali, the EU's MyCore project AflaSafe. And the report is clear, the funders didn't influence the study design or the results. Independent science.
Which is exactly what you want to see.
Okay, so moving from those toxins to diseases hitting the plant itself, let's talk stalk rots. We're drawing mainly from a Purdue extension document here.
Stalk rots, yeah. They're a big deal because they just weaken the plant structure. That can lead to lodging the stalks, breaking or bending over.
Which means lost yield, obviously.
Exactly. The Purdue guide focuses on two main ones, Diplodia stalk rot and Gibberella stalk rot. Different fungi, slightly different conditions favor them.
Diplodia stalk rot first, caused by Stenocarpella metis diplodia for short. Apparently it's been popping up more, and it's linked to Diplodia ear rot too.
That's right. And the conditions it likes are quite specific. A dry period before silking.
Silking being when the silks emerge from the ears.
Yes. And then followed by warm, wet weather after silking. That's the key window. The plant's most vulnerable then, post-silking.
And it attacks low down, the lower stalk, crown, roots.
Primarily, yes. And what's interesting, as the guide points out, is the fungus itself is actually pretty common, even on healthy-looking stalks. It just needs those favorable conditions to really cause disease.
So it's opportunistic, kind of waiting for the right moment.
You could put it that way, yeah. Now, Gibberella stalk rot is different, caused by Fusarium graminearum.
Which also causes Gibberella ear rot and Fusarium head blight in wheat and barley.
Correct. So that link means if you're rotating wheat and corn, especially in places like Indiana where Gibberella is common, you might see a higher risk. It also likes warm, wet conditions, but usually after pollination.
How do you spot Gibberella? Any telltale signs?
A key visual sign is these tiny, round, blue-black specks. They're called perithecia, right on the stalk surface, often near the first node above the ground. Apparently, you can just scrape them off with a fingernail. And if you split the stalk open, the inside, the pith, is often disintegrated, leaving just these stringy vascular bundles. And crucially, it often has a pink or reddish color. Figure 8 in the guide shows this quite well.
And how does it get in? The infection pathway.
Could be through the roots, or the stalk sheaths, or even the leaf sheaths. Several routes.
So, with these rots lurking, how can farmers check their fields before harvest to avoid major losses from lodging?
Scouting is crucial. The Purdue guide recommends doing this a few weeks after the corn hits physiological maturity. That's when you see the kernel black layer.
Right.
Walk the field maybe in a W pattern and check about 100 stalks randomly. Use either a pinch test or a push test.
Okay, pinch or push. How do those work?
Simple, really. Pitch test. Squeeze the stalk firmly between the lowest two inner nodes. If it collapses easily, it fails. Bad sign.
And the push test.
Push the top of the stalk sideways, maybe 8 inches or so at eye level, about a 30-degree angle. Then let go. If it doesn't spring right back to vertical, it fails that test.
And there's a threshold.
Yeah, the guide suggests if 10% or more of the stalks you check fail either test, you should seriously consider harvesting earlier. Even if the grain moisture is higher than ideal, get it out of the field before it falls over, then dry it down.
Makes sense. Better safe than sorry. The guide also flags general conditions favoring stalk rot's hot, wet, humid periods during the mid to late growth stages, V2 to R6.
And suggests some preventative ideas. Avoid overhead irrigation with stagnant water. Ensure good field drainage. Basically, try to minimize plant stress overall.
Right. Okay, so that covers specific stalk rots. Let's broaden out now to general strategies for managing fungal diseases in corn, using the AgriNews article as a guide.
Yeah, the AgriNews piece gives a good overview of the factors involved. Susceptibility depends on a mix of things, doesn't it?
Like what?
Well, the corn genetics, obviously.
Right.
Whether the seed was treated with fungicide, environmental conditions, huge factor when you plant, and of course, whether the pathogens themselves are present. Sometimes nematodes can play a role too, making plants more vulnerable.
And the threat starts early, right? As soon as the seed goes in the ground.
Absolutely. Seeds are immediately exposed to soil-borne fungi and bacteria. Some like it cool and wet, others prefer warmer soils.
And if seedlings get infected, what are the signs?
You might see damping off, or they just rot at the soil line and die. Or they could be stunted, yellow, maybe deformed. All signs you need to act fast if you see them.
The article flags some common fungal culprits. We see tar spot, northern cornleaf blight, gray leaf spot.
All big ones in recent years in many areas.
And while things like tillage, crop rotation, fungicides help, sometimes you need more direct management, depending on the year's weather.
True. The article lists several options. Starts with simple hygiene almost. Cleaning equipment between fields to avoid spreading spores.
Makes sense. Then choosing resistant varieties.
Super important. Look for those hybrids with higher resistance ratings for the diseases common in your area.
What else? Planting timing.
Planting for quick emergence helps. Less time for seedlings to be vulnerable in the soil. And using seed treated with a good systemic fungicide rated for the disease you expect.
Right, the seed treatment.
Balanced fertility is also key. Healthy plants are generally more resilient. And then there are in-furrow fungicides applied at planting.
But the article notes a potential downside there.
Yeah, the cost effectiveness. Might not pencil out if the disease pressure is only sporadic in your area. Worth considering, but maybe not always necessary.
Okay, what about spraying fungicides on the growing plant's foliar applications?
Generally, the recommendation is around or after tasseling. For varieties known to be susceptible, they suggest maybe spraying when symptoms first appear on the third leaf below the ear or higher, on about half the plants, right around tasseling.
And the best timing for a single spray.
Usually gives the biggest bang for your buck between VT, that's full tassel, and R1, which is silking. That window seems to be most effective for a single application.
The article really hammers home the need for regular scouting and correct diagnosis, doesn't it?
Absolutely critical. Because fungicides won't touch bacterial or viral infections. You have to know what you're fighting.
And it's not just if you spray, but when and how often. And using different types of fungicide.
Exactly. Using multiple modes of action is vital to slow down fungicide resistance development in the pathogens. That's a huge concern.
And the golden rule.
Always, always read and follow the label instructions for any product you use. Cannot stress that enough.
The article also mentions support from local experts like Channel Seed Pros for tailored advice and field evaluation tech.
Yeah, having that local expertise is invaluable. They know the conditions, the current disease pressure.
Especially as they note for Indiana, if planting gets delayed, something like tar spot might flare up more. So early scouting and local advice become even more important.
Makes perfect sense. And of course, the standard disclaimer. These are general recommendations. Always consult with experts for your specific situation.
Right. So we've covered spotting and managing. Now let's dig into the plant's own defenses, the genetics of resistance. There's a journal article that goes deep on this.
Yes, this is a fascinating area. The article points out that most maize diseases are actually caused by fungi, and the economic hit is massive yield loss, quality reduction, control costs.
Numbers involved.
Huge. And the major fungal threats. Foliar diseases hitting the leaves, smuts, and those stalk and ear rots we talked about.
Table 1 in that paper looks incredibly detailed.
It really is. It lists major diseases, the pathogens, scientific names, sometimes older names too, what type of organism it is, what the fungi feed on, where on the plant it shows up, global distribution, and importantly, examples of known resistance genes.
Wow. So for northern cornleaf blight, NCLB, caused by Exorahilum tursicum, they list a gene called ZEMYOKRLMT2.
That's one, yes.
Yeah.
And for southern cornleaf blight, SCLB, from Bipolaris matis, they mention ZMC-CoAOMT2. And that's just two examples. The table covers GLS, BLSB, rusts, smuts. It really highlights the diversity of both the threats and the plant's genetic toolkit.
And figure 1 gives visuals.
Yeah, pictures of the typical symptoms: NCLB lesions, GLS spots, SCLB lesions, rust pustules, smut galls, the look of gibberella stalk rot inside. It's a useful visual reference for what these diseases actually look like on the plant.
Let's take NCLB again. The article says it causes over 1% global yield loss annually and mentions big outbreaks in the U.S. and China.
Yeah, it's significant. And they discuss two types of resistance. Qualitative resistance, often strong resistance from single major genes like HT1, HT2, HT3, HTN1. These often give a specific chlorotic lesion reaction.
And they've actually found the gene behind HTN1.
Yes, that ZEMYOKRLK1 gene you mentioned. It acts as an immunity receptor. Interestingly, they found resistance linked to this gene correlated with lower levels of certain defense compounds called benzoxacinoids.
Huh, that's interesting.
It is. They also discuss how some HT genes are related and mention another recessive gene, HT4. But beyond these big single genes, there's also quantitative resistance, meaning resistance controlled by many genes, each having a smaller effect. These are scattered across all 10 maize chromosomes. They give Zimmerm's 6.3 as an example locus.
Okay, what about the soil-borne ones, smuts, head smut and common smut?
For head smut, caused by Sporosaurium earlianum, they've identified a key resistance region, a QTL called QHSR1, and pinpointed the causal gene, ZIMWAC.
Another ZIMWAC.
Seems to be an important family of genes. This one makes a protein on the cell wall that seems to stop the fungus spreading upwards in the plant. For common smut, Eustalogomates, they found resistance regions, but identifying the specific genes has proven trickier.
And stalk and ear rots? You mentioned resistance QTLs earlier.
Right, QRFG1 and QRFG2.
Yeah.
The article details the causal genes. For QRFG1, it's ZMCCT. Its activity is affected by whether a mobile DNA element, a transposon, is sitting in its control region, kind of like a switch.
Fascinating. And QRFG2?
That's linked to the Zimimox RP1 gene. This one seems to be involved in balancing growth hormones and defense compounds, conferring resistance to both gibberella stalk rot and Fusarium ear rot.
So one gene helping against two different rot issues.
It appears so. Shows how complex these genetic networks are.
The paper touches on bacterial resistance too, like Goss's wilt.
Briefly, mentions that the resistance QTLs found so far tend to have smaller individual effects compared to some of the major fungal resistance genes.
Where do breeders find these useful resistance genes?
The article highlights tropical maize lines, like one called Suwan1 for GLS resistance, also that hybrid P78599, which carried ZMCCT and ZMOX RP1. Even Teosinte, maize's wild ancestor, holds potential.
But some good genes are rare.
Yes, like certain beneficial versions or alleles of ZMCCT, or another one called ZMG-IHEL for maize rough dwarf disease resistance. That really underscores why collecting and evaluating diverse maize germplasm is so important.
You mentioned ZMCCT and ZMXRP1 having other effects too.
Right, cheiotropy. ZMCCT can affect sensitivity to day length, and ZMXRP1 influences root growth. These side effects have to be managed in breeding.
But they have successfully used these genes.
Yes, using marker-assisted selection, MAS. They've bred beneficial versions, like the H5 type of ZMCCT, into elite lines, improving stalk rot resistance without the negative side effects on day-length sensitivity, and even improving other agronomic traits. So it can be done.
And then there's genomic selection, GS.
A more recent approach. Instead of focusing on a few known genes, GS uses information from markers across the entire genome to predict the overall breeding value of a plant. It aims to accelerate genetic gain for complex traits like disease resistance.
But finding those quantitative resistance genes is still tough.
Very much so. It requires a whole battery of techniques: fine mapping, genome sequencing, looking at all the molecules, omics, using mutation libraries, even genome editing. It's a big challenge.
Okay, that genetic perspective is crucial. Let's shift now to a more recent review article from Frontiers. This one focuses specifically on 3 major fungal leaf diseases in Africa: northern leaf blight, gray leaf spot, and southern cornleaf blight.
Right. caused by Exorhalum tursicum, Cercosporazana, and by Polaris matus, respectively.
The article sets the scene by noting how modern farming practices like monoculture and conservation tillage can sometimes inadvertently help these pathogens.
Yes, saving soil is good, but disturbing it less can leave more infected crop residue on the surface, which is a source for some diseases.
It also reminds us of maize's global importance, that huge production figure, but points out that sub-Saharan Africa, while heavily reliant on maize for food, grows a smaller percentage of the world's total.
A critical context for understanding the impact there.
So let's look at the pathogens. Northern leaf blight, E. tursicum. What are the symptoms?
Typically grayish, long, elliptical lesions on the leaves. They start as small yellowish flecks. The fungus can get established pretty fast, 6 to 18 hours, and you see mature lesions within about 2 weeks under favorable conditions.
And the impact?
It infects the leaf epidermis and vascular tissue, reduces the photosynthetic area significantly, and can even lead to lodging. Yield losses can be severe. The review quotes figures from 30% up to a staggering 91%.
Wow, and it spreads easily.
Yes, via spores, conidia, carried by wind and rain splash, from old crop debris or existing infections.
The review also covers gray leaf spot, Cercosporazana, and southern cornleaf blight. By Polaris matus, they like similar conditions.
Yes, generally warm, 20-30 degrees C, and very humid, over 90% humidity. And again, significant yield loss potential from each 10% to 80%. The review makes a good point that we need to understand their combined impact better.
With global trade and climate change, accurate diagnosis is getting even more critical, the review argues.
Definitely. The traditional way is visual inspection in the field, using severity scales, like those 1 to 5 or 1 to 9 scales mentioned in Table 2.
But that has limitations.
Big time. It's subjective, it takes time, and crucially, the early symptoms, those little chlorotic spots shown in Figure 3, can look very similar for different diseases. Easy to misidentify.
So what are the alternatives? Digital imaging.
That's a promising area.
Right.
Using photos, regular RGB or hyperspectral from cameras, phones, drones, satellites, and then applying machine learning models to analyze the images for better, faster, more objective diagnosis.
Sounds high-tech.
It is. Things like SVMs, CNNs,
Yeah.
powerful tools. But there's also lab-based diagnosis.
Like growing the fungi, microscopy.
Exactly. Morphological diagnosis. Looking at spore shapes, for instance. Figure 4 shows how the hilum part of the spore differs between E. tursicum and B. matus. You can distinguish Cercospora species by their conidia too. But again, culturing takes time.
What about DNA methods?
Molecular identification. PCR. Sequencing specific DNA regions like the itis region. Table 3 lists primers used for these 3 fungi. These methods are fast, sensitive, specific. You don't need as much prior expertise.
They mentioned specific genes for distinguishing Cercospora types too.
Yes, CPR1 and CTB7. And looking at mating type markers, as in Table 4. There are also newer things like nanosensors and CRISPR being explored for diagnostics, but they're not quite ready for prime time for these specific pathogens yet.
The review also highlights the value of having the full genome sequences for these fungi. Table 5 lists them.
Absolutely crucial, especially with long-read sequencing, improving accuracy. Knowing the genome helps identify pathogenicity genes, like effectors those proteins fungi use to manipulate the host plant. Tools like Effector P help find these.
And knowing the genetics can lead to new control methods.
Potentially, yes. Like RNAi-based fungicides, which target specific fungal genes. A study on C. zana showed promise there.
But ultimately, the review stresses breeding for resistance as the best long-term strategy.
By far the most economical and eco-friendly approach. They discuss both types again. Qualitative, major genes, strong resistance, and quantitative, many minor genes, reducing severity.
We talked about the HT genes for NCLB.
Yes, the review details those again, HT1, HT2, HT3, HTN1, etc., and notes the protein HTN1 codes for quantitative resistance to all 3 NLB, GLS, SCLB. Lots of QTL mapping studies have been done, finding resistance loci across the genome.
Have they found specific genes for quantitative resistance too?
They mention one candidate gene cloned for GLS and SCLB resistance involved in lignin biosynthesis. And importantly, while some QTLs are environment-specific, others work more broadly and are being used in molecular breeding, specifically aiming to help African smallholder farmers.
The review also delves into the population genetics of these fungi in Africa. What do they find for E. tursicum, the NLB pathogen?
Quite high genetic diversity, especially compared to Europe. Sexual recombination seems to be a major driver of this diversity, even though the sexual stage itself is rarely observed. They note a need for more research on its population structure within Africa.
And Cercosporazana causing GLS.
Also high diversity in Africa. The population structure seems complex, likely resulting from multiple introductions, sexual recombination, gene flow, migration, and local adaptation. Interestingly, it seems CZAN is the sole cause of GLS in Africa. They haven't found the related CZ mates. Genome sequencing of African isolates is confirming genetic differences between populations.
What about bipolaris mates, SCLB?
Less information from Africa specifically. Studies elsewhere, like India, show high diversity with one race, race O, being dominant but still quite variable genetically. Again, sexual recombination seems important. Having the genome sequenced now should really help develop better markers to study its global populations.
So the conclusion is that these genetic insights hold promise for African breeders.
Huge potential, yes. But the review rightly points out the challenges. Breeders need access to diverse germplasm and research capacity. Resistance needs to work in local African environments. And they need to keep monitoring how the pathogen populations themselves are evolving.
Are there ways around those challenges? Germplasm access?
There are existing channels through national programs and NGOs, though shipping restrictions can be tricky. But importantly, there have already been successes in releasing stress-tolerant maize varieties in Africa, so there's a strong foundation to build upon.
Okay, so bringing this all together, we've really covered a lot of ground, from the hidden threat of aflatoxins to the very visible damage of stalk rots and leaf blights.
Yeah, a whole range of fungal challenges.
We've looked at how they're identified from just walking the field and looking to using sophisticated imaging and DNA techniques.
The diagnostic toolkit is expanding.
And we've explored management strategies, resistant varieties being key, but also cultural practices, smart fungicide use, and that continuous effort in breeding for better maize.
It really highlights the dynamic interplay, doesn't it, between the maize plant and these ever-evolving fungal pathogens. The science and breeding efforts are constantly trying to stay ahead, or at least keep pace.
It's definitely not static. New challenges, new solutions emerging all the time.
Absolutely. It's a continuous process.
Absolutely.
Yeah.
And maybe a final thought for our listeners to chew on. How might really understanding this microscopic battle, the fungi attacking and the plant defending at a genetic level, how might that understanding be fundamental to ensuring global food security and making agriculture more resilient in the future?
That's a great question. It connects the deep science directly to the food on our tables.
It really makes you think about the intricate connections between biology and our everyday existence. Thanks for joining us for this deep dive.